Hello and welcome to another Bio43 pre-lab video. This should be the final video you watch for the second week of lab. This video will walk you through an experiment to measure cilia function. You'll see an application for common biological equipment and techniques, learn about a technique you could apply when designing your own experiment, and later use this data to practice graphing and statistical analysis. Measuring cilia function directly can be a bit tricky, so we're going to rely on an indirect approach. Most of the cilia you can see on the tetrahymena are involved in motility, but there are also cilia surrounding the oral groove that are important for feeding. They sweep particles and media to the groove, which are then engulfed through a process called phagocytosis, forming a vacuole in the cell. The better the cilia function, the faster vacuoles will form. So we can use rate of vacuole formation as a measure of cilia activity. Let's look at an experiment that uses this approach. I read a paper that concluded glycerol inhibits cilia function in Tetrahymena piriformis. This led me to hypothesize that glycerol negatively affects Tetrahymena thermophilus cilia as well. We can set up a simple experiment to test this. We can have a control condition with Tetrahymena in a typical growth media, and an experimental condition of Tetrahymena in mostly the same conditions, but with 1% of the standard media replaced with glycerol. Based on our hypothesis, we would predict that within a given time frame, Tetrahymena in the 1% glycerol solution will form fewer vacuoles on average than Tetrahymena in standard media. So let's run the experiment. Back in the lab, I've collected all the materials we'll need: our Tetrahymena cells, some extra media, glycerol, India ink, and glutaraldehyde, which I'll explain the purpose for later, a compound microscope, along with slides and cover slips to let us view the tetrahymena, some microfuge tubes, one labeled for the control treatment and one for the experimental, micropipettes and tips for transferring liquids, and a timer, which in this case is my phone off screen. The experiment is simple to set up. I'll transfer 400 microliters of tetrahymena into both tubes. Then in the experimental tube, I'll add 500 microliters of 2% glycerol. But in the control tube, I'll just add 500 microliters of extra media. Then in both tubes, I'll add 100 microliters of 5% India ink. Invert a few times to mix, and now the experiment is started. While that runs, let me explain a little more about what I just did. First off, why the ink? The ink helps us observe the vacuole formation. India ink is really a suspension of tiny particles. The tetrahymena will engulf those particles like food and form dark vacuoles that will be easy to see. And by only counting the dark vacuoles, we know we are only counting the number of vacuoles that have formed since the ink was added. Now about the glycerol. I added 2% glycerol to the experimental tube, even though I said we wanted to use 1%. Well, the 500 microliters of glycerol I added ends up diluted to a final volume of 1 milliliter in the experiment tube. The final concentration is half of the stock concentration. So the concentration of glycerol in the experiment tube is indeed 1%. Keeping track of starting stock concentrations and final experimental concentrations is a tricky aspect of learning to design experiments. Finally, why did I bother adding 500 microliters of extra media to the control? That's all about keeping the control treatment as similar to the experimental treatment as possible. If the control tube only had 400 microliters of tetrahymena and 100 microliters of ink, it would be in a final volume of 500 microliters, and the India ink would be at a concentration of 1%. However, the experimental tube has a volume of 1 milliliter and an ink concentration of 0.5%. That difference could have a major effect on the experiment. So adding another 500 microliters of regular media to the control tube keeps the volume and ink concentration constant across both treatments. Now let's check in on the tetrahymena 5 minutes after we added the ink. Time to prepare slides. I'll just show the process with the control, but it will be the same for the experimental. Just take 30 microliters of the tetrahymena ink mixture and put it on a microscope slide. Then add 5 microliters of this 2% glutaraldehyde solution. Stir around with a micropipette tip to mix a little. And gently apply a cover slip. The glutaraldehyde is a fixative. Counting the vacuoles in active tetrahymena would be far too difficult. So the glutaraldehyde kills the tetrahymena in a way that preserves their structure, allowing us to count the number of vacuoles that formed up to the point the cells were exposed to the fixative. Glutaraldehyde is toxic to human cells as well, which is why I've been wearing gloves during this procedure. So now let's mount the slide with the glutaraldehyde on the microscope and find our tetrahymena with this nice microscope camera. Here's what it sees at first. We're on the lowest magnification, 40x, and can't see anything yet since we're not in focus. I'll start by adjusting the coarse focus, which will take a bit of time. These annoying lines will go away on higher magnifications. Eventually, we see our little guys. I'm going to adjust the position of this slide to try and center this group of 3 tetrahymena. Increasing the magnification narrows the field of view, so you always want to have your subject centered before you switch objectives. Now I'll go to 100x magnification. A little out of focus again, so I'll adjust with the fine focus knob. A better view here, but it would still be difficult to count the vacuoles. So I'll center that cluster again. And move on to 400x magnification. Now we'll be able to get good resolution on these vacuoles. Let's center these cells one more time. Play around with the fine focus here. It's important to remember the tetrahymena are 3-dimensional, so different parts will be in focus at different times. I'm trying to find a nice focus where all the vacuoles can be seen. This looks pretty good. Let me take a picture, crop the cell, and digitally enlarge on the screen. Now it's time to count the vacuoles. I would say there are 8 in this cell. Often there are some tricky calls to make. Zoomed in like this, it's easier to tell that this mass is 2 vacuoles right next to each other. This more faint spot is likely a vacuole out of focus, so we'll include that. I think this oddly shaped structure here is a vacuole in the process of forming, which I typically include in my counts. I don't count these tiny black dots that you often see at the wider end of the cell. The most important thing is to be consistent. If you use different standards for what counts as a vacuole for your control in your experiment, you're introducing a bias that can skew your results. And unless you're actively thinking about this and avoiding it, you could be doing it subconsciously. So that's it. That's what you'll be doing in lab this week. See you then.